Welcome back to the Deep Dive. Today we're really strapping in for a journey. We're going right to the edge of neuroscience, looking at something in the brain we thought we understood, but, well, maybe didn't.
Right, we're talking about astrocytes. You know, for the longest time, people just called them brain glue, simple support cells.
Exactly, just glue. But that story is, let's say, evolving rapidly. For decades, the idea was this thing called the tripartite synapse. You had the neuron sending the signal,
Right, presynaptic one, yeah.
the neuron receiving it, the postsynaptic one, and then this little bit of astrocyte, the PP, just kind of hanging around nearby, maybe cleaning up a bit.
Uh-huh, acting as a barrier or a mop, basically. That was the picture.
Well, our source material today is a really groundbreaking study that throws a lot of that into question. Our mission here is to dig into why that simple tripartite idea isn't enough anymore.
We need to shift perspective, move away from focusing on just one synapse at a time.
That synaptocentric view, right?
Exactly. Towards an astrocentric view, we need to understand the astrocyte's own structure, its own organization, and how that lets it manage, well, potentially whole circuits, not just single connections.
And it all starts with seeing things in a completely new way, doesn't it? The technology had to catch up first.
Oh, absolutely. To even conceive of this shift, the researchers needed a visual miracle, really. They had to uncover the astrocyte's internal logic, which turns out to be much, much more intricate than we thought.
Okay, let's start there then, with the visuals. How do you get from these blurry pictures of glue to, like, a detailed blueprint of this almost computational structure?
Well, they use some seriously advanced tech. It's called volumetric high-resolution electron microscopy, specifically FIB-SEM, focused ion beam scanning EM.
FIB-SEM. Okay.
And this allowed them to get images with an isotropic voxel size of just 4 nanometers.
4 nanometers. That's hard to even wrap your head around. What kind of context can we give for that? It's like mapping a city down to the individual bricks.
Maybe even smaller.
Yeah.
Like mapping the wiring inside every single light switch in a giant skyscraper. It's that level of detail, and it was absolutely essential.
Why? What were they trying to see that needed that kind of, well, zoom?
Because the key parts of the astrocyte at the synapse are incredibly thin and really folded up, convoluted. So this resolution let them clearly define 2 main parts of the astrocyte structure, new synapses.
Okay, lay them out for us.
First, you have the shafts. Think of these as the larger branches coming off the main cell body, bigger than 250 nanometers in diameter, generally.
The main highways.
Kind of, yeah. They're the structural backbone, and they're packed with the cell's heavy machinery mitochondria for energy, and the complex endoplasmic reticulum, the CER, for calcium storage and protein handling.
Got it. Shafts are the trunks and big branches. What about the parts actually touching the synapses, the business end, so to speak?
Right, those are the leaflets. These are super thin, branching, folding sheets like delicate lamellae. They're defined as being 250 nanometers or thinner.
Okay, thin sheets.
But the key identifier, the thing that really tells you you're looking at a leaflet, is this sudden, massive jump, about tenfold in the surface-to-volume ratio, the TEX-DR, compared to the shafts they branch off from.
Whoa, okay, SVR, surface-to-volume ratio.
Yeah.
For those of us not doing cell geometry daily, why is a high SVR important here? What does that tell us?
It tells you what it's built for, interaction, maximum surface area crammed into the smallest possible volume. Think about crumpling up a piece of paper, same amount of paper, but it takes up less space and has way more surfaces touching.
Ah, I see. So the leaflet is crumpled up specifically to touch and wrap around as many synapses as possible in that little bit of space.
Precisely. It maximizes the contact area.
Okay, that explains what these thin, high surface area leaflets. But you said the breakthrough was also about how they connect. The old model was like 1 astrocyte finger per synapse, right?
Yeah, roughly.
So what did the 4 nanometer view show? Are they separate fingers?
No. Not separate at all, actually. This was a huge finding. The leaflets connect to each other. They use these specialized structures, gap junctions.
Gap junctions. Okay, I know those. They let stuff pass directly between cells.
Exactly. These gap junctions contain a protein called connexin 43, and they form direct bridges, linking one leaflet to its neighbor, or sometimes linking a leaflet back to a shaft. This creates these large interconnected networks.
So they're not individual units. They form domains. Like a shared network space.
Yes, the researchers call them leaflet domains. Because of the gap junctions, the cytoplasm inside these connected leaflets is continuous. It's like one big shared internal space. They're working together as a single functional unit, a syncytium.
That completely changes things if they're all connected like that. What does it mean for the synapses they touch? Is the old tripartite synapse model even relevant?
That's the crucial question the study raises. They found that, yeah, about 98% of synapses are contacted by leaflets, so the astrocyte is definitely there. But only about 10% of those contacts fit the old model one synapse, one little leaflet process.
Only 10%. So what about the other 90%?
The vast majority, 90% of synapses, were different. They were clustered together, all nestled within or contacted by the same leaflet or within one of these large interconnected leaflet domains.
So instead of one-on-one coverage, it's more like the astrocyte is covering a whole group of synapses together within this network domain.
Exactly. It's not a collection of individual tripartite synapses anymore. It's more like a tripartite synaptic network. That's the term they propose. The astrocyte structure physically links neighboring synapses together.
Wow. Okay, if only 1 in 10 fits the old model, why did we rely on it for so long? Was it just wrong?
It wasn't necessarily wrong. It was just incomplete, limited by what we could see. Standard light microscopy just couldn't resolve these incredibly thin leaflets, sometimes only 15 nanometers thick, or the gap junctions connecting them. We saw the astrocyte nearby, but missed this whole layer of organization.
So the technology revealed the true architecture is built for group coordination, not just individual monitoring. The astrocyte forces synapses to be part of a local network.
That seems to be the implication, yes.
Okay, let's recap this first part quickly. We've got this new view thanks to super high-res EM. Astrocytes at the synapse form incredibly thin leaflets with a very high surface-to-volume ratio for maximum contact.
Right, and these leaflets aren't isolated. They connect via connexin 43 gap junctions to form large interconnected leaflet domains.
Which means the classic one astrocyte process per synapse model only holds for about 10% of cases. The reality for 90% is this clustered tripartite synaptic network.
Precisely. The structure itself implies coordination.
Okay, structure established. We have the chassis, these high surface area leaflets. We have the network, the gap junctions. Now, every structure needs an engine, right? What powers the communication within these new computational domains?
Right, the engine. And to find that, you have to look inside the leaflets, at the organelles, and maybe the biggest surprise here is what's missing.
Missing? What's not there?
Well, for starters, mitochondria, the cell's power plants, are almost completely excluded from the leaflets themselves. They usually stop right at the boundary where the leaflet branches off the shaft.
Huh. Okay, no local power stations. What else?
The complex ER, the CER, which is this big network that stores loads of calcium in the shafts and cell body, that's also largely absent from the leaflets.
So the main power lines and the main calcium tanks are back in the highway shafts, not out in these fine contact leaflets. How does the leaflet manage rapid local signaling then? Where does the calcium come from?
It comes from specialized, tiny local stores. Inside the leaflets, they found these distinct, minuscule organelles. Isolated ER saccules, or IER.
Isolated ER, like little fragments.
Exactly. Tiny fragments, typically only about 50 nanometers across. That's about 100 times smaller in volume than the big CER network back in the shafts.
Wow. Like little emergency calcium packs scattered throughout the leaflet domain.
That's a great way to think about it. Localized, fast-response calcium packets placed strategically right where the action is.
Ultimate functional specialization. Okay, why are these little IER bits so important? What makes them the engine?
Because they contain the machinery needed for calcium signaling. They express IP3 receptors. These are the channels, the gates, that release calcium when they bind to a molecule called text IP33.
And text IP3 is generated by certain types of synaptic activity, right?
Correct. Specifically, GQ protein-coupled receptors activated at the synapse produce text IP33. And crucially, these little IER saccules are positioned perfectly to detect it.
How close are they?
On average, just about 607 nanometers away from the synaptic cleft itself.
600 nanometers. That sounds tiny. Is that close enough for the text IP3 signal to travel quickly from the synapse to the IER?
Yes, it's well within the expected diffusion distance for text IP33. The signal can jump almost instantly from the active synapse to trigger calcium release from these nearby saccules.
Okay, so the neuron talks, text IP3 is made, it hits a nearby IER saccule, and boom, calcium is released inside the leaflet. But you mentioned these leaflet-formed domains. Can the signal spread?
It seems like it's set up for that too. The average distance between these IER structures within a leaflet is about 573 nanometers.
Also very close.
Right. And that distance is compatible with something called calcium-induced calcium release, CICR. It means when one saccule releases calcium, that calcium can diffuse to the next saccule and trigger it to release calcium too.
Ah, like a chain reaction. One spark ignites the next, propagating the signal across the whole connected domain.
Potentially, yes. So the IER can both initiate the signal locally in response to a synapse, and then help propagate that signal throughout the network formed by the gap junctions.
So the neuron provides the trigger, but the astrocyte's own structure and these specialized IER engines amplify and distribute the signal across its domain. Okay, we've got the structure, we've got the engine. How did they actually watch this system running in real time? How do you image calcium in these tiny specific compartments?
Yeah, that was tricky. They used two-photon calcium imaging, which lets you look deep into tissue. But how do you target the leaflet specifically? They used a really clever approach, kind of like negative staining.
Negative staining, how does that work?
Well, they knew leaflets don't have mitochondria, right?
They're excluded.
So they used a fluorescent marker that specifically labels mitochondria. Then they looked for the dark, non-fluorescent areas right next to known synapses, which they marked with another tag, PSD95. Those dark spots, lacking mitochondria but right next to synapses, were inferred to be the leaflets.
Huh, that's smart. Define the space by what's not there. Did that indirect method work? Was it reliable?
It seems so, especially because the high-res EM structural data backed it up, showing mitochondria absent in those thin synapse wrapping processes, and the functional results were really clear.
Okay, what did they see in those dark spot leaflet regions?
They saw calcium signals, TEX2+ activity, that was very clearly segregated from what was happening in the nearby mitochondria-filled shafts. And crucially, the leaflet calcium signals were strongly neuronal activity dependent.
Meaning they only happened when neurons were firing.
Pretty much. When they added drugs that block synaptic transmission like TTX to block sodium channels or VGZC antagonists to block calcium channels needed for transmitter, released the calcium activity in the leaflets dropped by a whopping 95%.
95%. So the leaflet is almost purely listening to the neurons. Its calcium signals are almost entirely driven by synaptic input.
That's what it looks like, yes. It's the dedicated synaptic listener.
And what about the shafts, the areas with mitochondria?
Interestingly, the calcium activity in those neighboring shaft regions was not significantly suppressed by the synaptic blockers.
Ah, okay. So the shafts are doing their own thing, calcium-wise, probably related to metabolism or other internal processes, independent of the immediate synaptic chatter.
Seems likely. This really reinforces the idea of the leaflet as this specialized computational compartment specifically tuned to synaptic activity.
Okay, that's a huge functional confirmation. Now let's get into a really interesting molecular detail. The signaling needs those IP3 receptors, the text IP3 text. But for anyone who follows astrocyte research, the identity of which IP3R is involved has been, well, a bit of a mess, right?
Oh, absolutely. A long-standing debate. For years, the main suspect, especially for widespread calcium waves in astrocytes, was thought to be IP3 receptor type 2.
Right, text inhibitor gets a lot of attention. But there have always been these nagging reports, these observations of calcium signals that still happen even when text IP3 text is removed, signals that seemed independent of it.
Exactly. There was this puzzle of text IP3 dependent signaling. This study provides a really compelling answer, at least for the specific astrocytes. They looked at those in the dentate gyrus molecular layer, the DGML.
Okay, what did they find?
First, they confirmed the signals are IP3 are dependent. When they used genetic tools to knock out all three types of IP3Rs, types 1, 2, and 3 in astrocytes, the leaflet calcium activity dropped by 85%. So IP3Rs are definitely required.
Okay, confirms the mechanism, but which one?
Here's the twist. When they deleted only TEX-RE22, the one everyone focused on, it did not significantly change the synaptically driven calcium activity in the leaflets.
Wow. So the main suspect, TEX-IP3, is not the primary driver for these specific synapse-linked calcium signals in the leaflets.
It appears not in this context. And they knew from gene expression data that these particular DGML astrocytes make plenty of IP3 receptor type 1, TEX-IP3, TEX-3 on type 2, but almost no type 3.
So if it's not type 2 and type 3 isn't really there.
Exactly. It strongly points to TEX-IP3, TEX-1 on as being the main trigger for calcium release within these specialized leaflet domains in response to synaptic activity.
That's a fundamental clarification. It suggests the structural specialization of the leaflet comes with a specific molecular toolkit, using TEXT-IP3 texton for this job. It resolves a big piece of that old debate.
It certainly provides strong evidence for TEXT-IP3 texton in this role, yes. A very neat structure function linked down to the molecular level.
Okay, let's pause again. Quick recap. The engine involves tiny IER saccules inside the leaflets. These are packed with IP3 receptors to release calcium.
And the calcium signals in these leaflets are almost entirely dependent on neuronal activity.
And crucially, the key molecular player triggering this release seems to be text IP3, text 1, not the long-assumed text IP3, RGB2, at least in these specific synapses.
That's the evidence, yes.
All right. Structure, engine, fuel. Now let's talk about the computation. The astrocyte, through these leaflet domains, is acting like a network coordinator. How does that actually manifest in the calcium signals themselves? What does the output look like?
Well, when they analyzed the patterns of calcium signals within the leaflets, they saw they weren't all the same. They fell into two main types.
Yeah.
About 30% of the calcium events they detected were what they called mono-originated. These were relatively small, short-lasting signals that seemed to start from just one point within the leaflet domain.
Mono-originated.
Ah.
Single source. Like maybe representing input from just one synapse firing.
That's the likely interpretation, yes. A single synaptic input hitting the domain triggers a localized, brief calcium response. That's the basic unit of listening, you could say.
Okay, but that's only 30%. What about the other 70%? You said they were different.
Yeah, the majority, 70%, were much more interesting. They called these multi-originated.
Multi-originated, meaning?
Meaning these events clearly started from two or more distinct places within the leaflet domain, sometimes at slightly different times. But then the signals would spread and merge together into one larger combined event.
Ah, so not just one input, but multiple inputs being registered and somehow combined within the same domain.
Exactly. And these multi-originated events were significantly different in character. They were, on average, 3 times larger in amplitude and lasted twice as long as the mono-originated ones.
3 times bigger, twice as long. That suggests something more than simple addition, maybe?
Yeah.
Like the domain is integrating these multiple inputs over space and time.
That's precisely the idea. It's not just counting single pings. It's taking signals that might be arriving asynchronously, say, axon 1 fires, then maybe 150 milliseconds later, axon 2 fires nearby, both contacting the same leaflet domain, and the astrocyte domain seems to combine this information.
Synthesizing inputs from potentially independent circuits into one larger, more prolonged signal. That sounds like computation.
It really does. And they got direct evidence for this integration. They used a really elegant dual-color imaging experiment.
Okay, how did that work?
They used one fluorescent sensor for calcium inside the astrocyte, GCAMP6, green, and another fluorescent sensor for calcium inside the nearby axons, ArcNMP3, red. So they could simultaneously watch the astrocyte domain and the activity of the individual neuronal inputs contacting it.
Wow, okay, so they could see if axon A firing, then axon B firing actually led to one of those big multi-originated events in the astrocyte.
Yes, and they showed exactly that. They found instances where a multi-originated calcium event in a single leaflet domain clearly reflected the combined asynchronous firing patterns of 2 separate neighboring axons, axon 1 and axon 2, that were both synapsing within that domain.
That's direct proof of integration. The astrocyte isn't just listening to one neuron. Its domain structure allows it to listen to multiple neurons, potentially from different circuits, and combine their messages over time.
Precisely. The structure enables the function. That complex organization we talked about, multiple synapses from potentially independent axons or dendrites, all converging within one gap junction connected leaflet domain that provides the physical platform for this spatiotemporal calcium integration.
So astrocytes are performing a type of computation that's distinct from what neurons do. Neurons integrate inputs to decide whether to fire an action potential. Astrocytes seem to be integrating inputs to coordinate activity across groups of synapses, across different circuits.
That's the picture emerging, yes.
Yeah.
They're acting as integrators, coordinating synaptic ensembles, potentially operating across different spatial and temporal scales than neurons typically do.
This is really a paradigm shift, moving from passive glue to active participant at one synapse to now this network coordinator, this computational integrator layer.
Yeah. Okay, let's try to wrap this up. Let's hit the key takeaways from this deep dive one more time.
Okay. So structurally, the astrocyte interface at the synapse isn't simple processes. It's dominated by these incredibly thin, high surface area leaflets. And these leaflets don't work alone. They are interconnected via connexin 43 gap junctions to form large leaflet domains.
Right, which means the old tripartite synapse model is insufficient. About 90% of synapses are part of a coordinated tripartite synaptic network clustered within these domains.
Functionally, the signaling engine relies on tiny, localized IER saccules within the leaflets, which release calcium.
And the key trigger for that release, driven by synaptic activity, appears to be primarily the text IP3 text R1 receptor, not text IP3 text, in these parasynaptic domains.
And the computational output. This structure-function system generates complex, multi-originated calcium signals representing about 70% of events that physically integrate asynchronous inputs from multiple converging neurons or circuits.
Yeah, so astrocytes are acting like calcium buffers, sure, but they're also accumulators of synaptic information over time and space. They gather this information to generate these integrated signals that likely influence how those synaptic ensembles behave. It's way beyond glue, and even refines the tripartite synapse idea significantly.
It's a whole other layer of information processing we were largely blind to. Astrocytes as circuit integrators.
Wow, it really changes the perspective.
It absolutely does. Which brings us to our final thought, something for you, the listener, to mull over. We've seen how this intricate leaflet domain structure allows astrocytes to perform a kind of computation, integrating information from independent sources. So here's the question. What if diseases that involve disordered thinking or information processing, things like, say, schizophrenia, or maybe even aspects of Alzheimer's, what if they aren't just problems with neurons, but also problems with this astrocytic integration layer? What if the astrocytes are failing to correctly combine or filter these signals from different circuits?
That's a fascinating and potentially crucial area for future research. If this integration is fundamental, its failure could have profound consequences.
And remember, we only saw this level of detail because of technology like 4-nanometer FIB-SEM. It makes you wonder, what other specialized cellular compartments, what other hidden computational structures might still be waiting to be discovered in other parts of the brain or even other cell types once our eyes get sharp enough?
The revolution is likely just beginning.
So think about that hidden complexity, that computational power nestled right next to the synapse. The next time you hear astrocytes dismissed as just brain glue or even simply part of the tripartite synapse, there's clearly a lot more going on.